So far, we have looked at the development of T lymphocytes and the cellular components of the adaptive immune response. In this video, we are going to focus on the other main cell of adaptive immunity, the B lymphocyte, and the soluble or humoral mediators they release, antibodies. We produce millions of B lymphocytes in the bone marrow every day, so we will look briefly at how the B cells develop in a tightly regulated process, and then the functional roles of these cells in adaptive immunity. B cells develop and mature in the bone marrow in humans. The bone marrow has a series of niches where the hematopoietic stem cells reside, and these can interact with bone marrow stromal cells, and the common lymphoid progenitors that then go on to become B lymphocytes are heavily influenced by the chemokine CXCL12 and interleukin 7 as they move from a pre-pro B cell to an immature B cell. And these cytokines and chemokines increase the cell survival, enhance proliferation, as well as drive the rearrangement of genetic material to create a diverse range of B cell receptors. Similar to the process that T cells undergo, the immature B cells have to pass a series of checkpoints. The first being the expression of a functional pre-B cell receptor at the pre-B cell stage, and any that do not are removed from the repertoire. A second check is then made at the immature B cell stage to ensure that that pre-BCR has successfully become a functional B cell receptor, and again, if not successful, any nascent cells die by apoptosis. At this stage, the B cell receptor must be tested to ensure it does not recognize self-peptides. BCRs that do recognize autoantigens can be deleted from the repertoire in the bone marrow through central tolerance, or alternatively, the BCR can reactivate the genes responsible for the genetic rearrangement and be edited. As with T cells, some autoreactive B lymphocytes may escape screening, but these will either never encounter their cognate peptide MHC complex or not receive appropriate co-stimulation and become anergic. Those cells that do not recognize a self-peptide can leave the bone marrow and move into the periphery. The final stage of B cell maturation happens outside of the bone marrow. The B lymphocytes that have made it this far leave the bone marrow and travel through the circulation to the spleen, which is one of the secondary lymphoid organs, and is this fist-sized organ sitting around the stomach region. The region of the spleen that we are interested in here is the B follicle. The follicle sits just outside the T cell zone, but inside the marginal zone, and we find B cells spread across the follicle and into this marginal zone. Immature B cells that recognize self-antigens with high affinity are removed by apoptosis before they even reach the follicle by negative selection. And once in the follicle, they undergo a process similar to positive selection, where the B cell needs to receive a combination of signals in order to survive. So just to summarize this, the process of B cell maturation in the bone marrow allows for the regulation of the BCR construct, in much the same way as a T cell forms the T cell receptor. It ensures that each B cell has only one specificity, so it can only recognize one epitope. Those B cells that react against self-antigens are removed by negative selection, and then functional B cells are allowed to pass into the periphery to continue their journey into the lymphoid tissues, where they wait to encounter a circulating pathogen or instructions from dendritic cells and T cells to generate an antigen-specific response. The bone marrow also provides a site for antibody production, because it's important to know that although B cells cannot become activated in the bone marrow, this happens in the lymphoid organs. Once a B cell has become activated, it may return to the bone marrow niche as a plasma cell secreting antibodies. The primary way that B cells become activated is through a T cell dependent route. This is where a CD4 positive T cell has been activated by a dendritic cell. It has co-stimulatory molecules, and it uses its T cell receptor to interact with the MHC class II molecule on the B cell, presenting a complementary peptide. Alongside this, T cells are secreting cytokines that are specific to the eliciting agent, and the combination of these three signals helps instruct the B cell as to the type of antibody it needs to produce. Alternatively, the B cell, being an antigen-presenting cell in its own right, can cross-link an antigen on its surface, internalize it, and present a peptide to a complementary T helper cell, and then receive the signals back from the surface receptors and soluble cytokines that are released from the CD4 positive cell, and it essentially gives the B cell permission to activate. From here, the B cell can undergo one of three fates. It can become a plasma cell that secretes antibodies, it can become an IgM-bearing memory cell, or it can enter the germinal centres in the lymphoid tissues and undergo an extraordinary differentiation process. B cells can encounter antigens naturally via a couple of different routes. Blood-borne pathogens are carried in the circulation to the spleen, while antigens in tissue spaces are carried in lymph and/or presented by dendritic cells or macrophages in the lymph nodes, with the B cell sequestering the antigen from these cells and then presenting it to the CD4 positive T cells to provide that required activation signal. The regions within these organs that provide the structure for the B cells to carry out their roles are the follicles and germinal centres, with marginal zone B cells playing an important role in antigen transport and presentation, particularly in the spleen. Now that we have a basic idea of how B cells become activated, what is the functional consequence? Well, the primary output is the production of antibodies. So we need to take a look at antibody structure and function to better understand how this molecule contributes to the immune response. Antibodies are Y-shaped glycoproteins with a number of subunits and features that you should be aware of. In this schematic, you can clearly see the Y-shaped structure, and that structure is made up of two identical light chains and two identical heavy chains. Both the light and heavy chain have constant regions, with one constant region shown here on the light chain, labelled Cl, and in this example, three constant domains in the heavy chain, labelled CH1, CH2, and CH3. The number of constant heavy domains can vary between antibody classes, with IgE and IgM having 4 constant domains, and IgA, IgD, and IgG having 3. We will discuss these different classes and their functions a little later in the video. At the N-terminal end of the chains, we have the variable region of the light chain, and this corresponds to a variable region on the heavy chain as well. The two variable regions of the heavy and light chain result in the antigen binding site, and it is the hypervariability of this region that allows for such diverse antibody specificity. Antibodies, despite their extensive covalent and non-covalent bonding, are not rigid structures, and the hinge region, where the arms of the Y join the stem, allow for a degree of flexibility that helps antibodies bind to their targets. If we look at the crystal structure of an antibody, it becomes quite apparent why the name immunoglobulin is appropriate, with these thick globular structures formed by the tight folding of the amino acid sequence linked with the flexible hinge region. So in this image here, we have the two heavy chains coloured in light blue and yellow, and the two light chains in dark blue and green. Humans can make 5 different classes of antibodies, and although there are similarities between all the classes, there are also a few differences, as you can see from the representations here. So the structure we talked about previously was based on IgG, and this has a very similar structure to IgD. IgE also takes on a classic monomeric Y shape, but you will notice that it has an extra constant domain on the heavy chain. So it has 4 domains making up the constant region of the heavy chain, compared to the 3 in IgG and IgD. IgA typically occurs as what we call a dimer, so it has 2 of these antibody structures joined at the foot end of the molecule, or what we call the Fc region of the molecule, by a structure called the J chain, or the joining chain. And then this dimer is further stabilised by the presence of a secretory component that spans the 2 molecules. IgM can exist as a monomer on the cell surface, and it is often the structure we refer to as the B-cell receptor on naive B cells. But in its soluble form, it exists as this pentameric structure, so 5 immunoglobulin subunits joined at the Fc end of the molecule by the J chain. Taking a look at each of these classes of antibody individually, Immunoglobulin M, or IgM, typically makes up about 5 to 10% of the total serum antibody. So remember, antibody can circulate in the bloodstream or can be expressed on the surface of B cells as its B cell receptor. And IgM as a monomer is often expressed as the antigen receptor on naive B cells, so B cells that have matured but have not yet encountered their antigen. It is typically the first class of antibody released during the primary immune response. So if we revisit our overview of antibody production during the primary and secondary immune responses, then the primary response is due to the secretion of IgM from activated plasma cells. We also get IgM as part of the secondary immune response, but it is a smaller component compared to the other classes, and this shift is driven by the CD4 T helper cells. IgM is also a potent activator of the complement system, so it plays a key role in the initiation of the classical pathway of complement, resulting in pathogen lysis. The most common form of immunoglobulin is IgG, which is a monomer and represents about 80% of free antibody in the plasma. There are 4 distinct subclasses of IgG, which makes it a little bit more complex to talk about. And these are called IgG1, IgG2, IgG3, and IgG4. So very easy to remember, and also a good indication of their relative abundance, because IgG1 is the most abundant and IgG4 is the least abundant. The subclasses have slightly different effects due to changes in the Fc region of the molecule, and these bind to cells via different Fc receptors. IgG1 and IgG3 are excellent opsonins, so they coat pathogens and help in the process of phagocytosis by neutrophils and macrophages. IgG3 is a potent activator of the classical pathway of the complement system, whilst IgG1, IgG3 and IgG4 have important roles in crossing the placenta from mother to fetus to convey passive immunity to the newborn. IgG has an important role in the secondary immune response on re-exposure to a pathogen. From the most abundant to one of the least abundant, IgD, which makes up less than 1% of the total serum immunoglobulin, but is co-expressed on the surface of naive mature B cells, where it seems to provide additional activation signals to the B cell in response to specific types of immunogens. Until recently, immunologists have been stumped as to the biological effector function of soluble IgD, but very recently a potential role in respiratory tract defense has been proposed, by driving the activation of basophils and mast cells to produce antimicrobial peptides. There is therefore a possibility that IgD may also contribute to allergy, as these cell types are very important in the allergic response. IgA only makes up about 10% of the total serum antibody titer, but it is the most predominant form in our external secretions, so for example our tears, saliva, or if a woman is lactating, their breast milk. It plays a central role in barrier immunity, so the airways, digestive and urogenitory tract. Compared to other classes of antibody, it is produced in far higher quantities, as much is excreted from the body by the flushing effects at these mucosal surfaces. IgA is very effective at protecting these surfaces because it can bind to extracellular bacteria and viruses and prevent them from interacting with epithelial cells. Because IgA is a dimer and the two Fc regions are joined by the J chain, they therefore cannot interact with Fc receptors on our mucosal cells, so it encourages the removal of pathogens away from our barrier surfaces. Last but no means least is IgE, which is by far the least abundant form of immunoglobulin under normal circumstances, but becomes elevated in individuals suffering from a parasitic infection or from those that suffer from allergic diseases such as asthma, allergic rhinitis, which is also called hay fever, or eczema. IgE activates blood-borne basophils and tissue-resident mast cells, with the antigen causing cross-linking of receptors, which results in the release of pharmacologically active mediators such as histamine via the process of degranulation, and this causes the clinical inflammatory response that we identify as allergy. And this is the process here being shown in the cartoon, with the antigen, which in this case is probably an allergen like pollen or animal dander, binding to and cross-linking the IgE on the surface of the mast cell, which becomes activated and degranulates, releasing these mediators and resulting in the clinical signs of allergy. Antibodies have a broad range of physiological effects, and to revisit some of these, antibodies can neutralize threats directly, so they can bind to soluble antigens or microbes and prevent them from binding to a cell, and therefore prevent any detrimental effects. They can opsonize pathogens, so the variable or Fab region of the antibody can bind to epitopes on the surface of the pathogen, and then the Fc region, the foot region of the Y, can bind to receptors on the surface of our phagocytes and allow them to engulf the pathogen. Antibodies, especially IgA and IgM, because of their multivalent nature, are able to cross-link multiple epitopes on pathogens, and therefore limit their movement, as well as flag them for destruction. IgG, especially IgG3 and IgM, are potent activators of the classical pathway of the complement system, whilst antibody release can drive the recruitment of innate immune cells to the site of infection. They also play a role in antibody-dependent cell-mediated cytotoxicity, or ADCC, whereby antibodies bind to target cells, for example a virally infected cell or a tumour cell, and allows various cells of the immune system, like natural killer cells or cytotoxic T cells, to bind to the Fc region and release their cytotoxic cargo onto that target cell. Now, if we revisit the basic structure of an antibody, we can draw a line of symmetry down the middle of the molecule, along the long axis of the Y, and this is because each side of the molecule is a mirror image of the other. So the amino acid sequence in the two heavy chains are identical, and the two amino acid sequences in the light chains are identical. And this means that the antigen binding site of any naturally occurring antibody is a paratrope. They will bind to exactly the same epitopes. The specificity is encoded within regions of the variable domain called complementarity determinant regions, which are also sometimes called hypervariable regions, and there are 3 in each of the heavy and light chains. These are called CDR1, CDR2, and CDR3, with CDR3 being the most variable. The regions between the hypervariable domains are somewhat less variable, and we call these the framework regions. The amino acid sequences in the variable regions varies dramatically between antibodies and confers a unique specificity of that individual antibody. If we align these regions to the crystal structure of an antibody, we see that these complementarity determinant regions cluster together at the tips of the heavy and light chains in the variable domains. When the heavy and light chains of an antibody are brought together, the variable regions of the heavy and light chains are paired at the tips of the arms. And this forms the antigen binding site in the Fab region. And this is totally unique to that antibody and allows it to bind with very high specificity to a complementary epitope on an antigen. So as you can see from this representation, only the first antibody can bind to the triangular epitope. Only the middle antibody, the star, and the pentagon can only bind to the last antibody. And these antibodies do not recognize the other shapes because they are not complementary to the binding site formed by these hypervariable regions. Now, something to consider is that antibodies are only recognizing a very small part of an antigen, and this means that multiple different antibodies from multiple different B cells can bind to different parts of the antigen, and these regions are the epitopes that we have spoken about, and sometimes referred to as the antigen determinant regions. What we can see in the diagram are 2 different antibodies binding to individual regions of the antigen, and those regions highlighted in blue and red are these epitopes on the antigen. We also have different types of epitopes. So the red epitope is what we call a discontinuous epitope or a conformational epitope, because it arises from a non-linear sequence of amino acids brought together by the 3-dimensional structure of the antigen. In contrast, the blue epitope is a continuous or linear epitope because it is made up of amino acids that appear in sequence in the molecule. Moving from the variable region to the constant region, we need to discuss class switching, which is a mechanism by which B cells can change their production of antibodies from one class to another. For example, from IgM in the primary immune response to IgG in a secondary immune response. You may also see this called isotype switching or class switch recombination, but all these names describe the same process, which is the switching of the constant region of the heavy chain, while the variable region of the heavy chain and the entire light chain remain the same. So the antigen specificity is retained, but the Fc region is altered, which can change the effector molecules or cells that can interact with the antibody. Importantly, this process of class switching only occurs after the naive B cells have encountered their antigen for the first time, and is heavily influenced by the cytokines that are released by the CD4 positive T helper cells, as specific classes of antibodies must match the response to the eliciting agent, and different classes will bring in different immune functions. The class or isotype that an antibody belongs to is determined by the heavy chain, and we denote this with the Greek letters gamma, mu, alpha, epsilon, and delta to represent IgG, M, A, E, and D respectively. We have already briefly touched on the fact that some classes have different subunits that provide further diversification or effector function, for example IgG. And we have receptors that are expressed on the surface of cells that can recognize these different constant regions. If we return to our example of allergy, then on first encounter with a potential allergen, our naive B cells that are producing IgM will class switch to produce IgG, and many people have IgG antibodies to common allergens, so pollen, animal dander, dust mites, etc. But they are not allergic. In allergic sufferers, cytokines that come from a CD4 positive T cell with a Th2 phenotype, so for example IL-4 and IL-5, will induce class switching so that the B cells start to produce IgE, and this binds to mast cells, basophils and eosinophils, and through degranulation triggers the clinical signs of allergy. So this really gives you an idea of the importance in the crosstalk between the different cells of the immune system. So this has been a summary of the journey that the B cell takes from being a naive B cell towards becoming a plasma cell or a memory cell. Naive B cells, when they encounter their cognate antigen, migrate to the dark zone of the germinal centre and undergo clonal expansion. We haven't yet talked about somatic hypermutation, but this is a further way that B cells can refine the effectiveness of the antibodies they produce, and we will discuss this in a future video. But in short, if a change is beneficial, then the B cell survives. If it is detrimental, then the B cell undergoes apoptosis. The activated B cell can then integrate with follicular dendritic cells and CD4 positive helper T cells, and will undergo class switching, with the resultant B cell becoming either an antigen-producing B cell or a long-lived memory cell that provides immune memory. So that has been a summary of the role of B cells and the soluble mediators they produce, antibodies. Antibodies have numerous roles within the immune system, bridging the innate and adaptive arms of the response, and B cells are a fundamental component of the adaptive response, contributing to the initial response and to immune memory, ensuring that we can neutralize threats effectively and restore tissue homeostasis as quickly as possible if we ever encounter the same challenge again.